Inside every dog and cat lives an invisible ecosystem more complex than a rainforest. 10 trillion bacteria outnumbering their own cells by 10 to 1, governing digestion, immunity, and even behavior. When this ecosystem thrives, health follows. Today, we are exploring the gut microbiome, the microscopic universe that determines whether your patients suffer from chronic diarrhea, inflammatory bowel disease, or maintain robust gastrointestinal health throughout their lives. I am Dr. Ashok Patnaik, former scientist from the Indian Veterinary Research Institute. 3 decades of companion animal nutrition research have taught me that gut is not just a digestive organ, it is the command center for immunity, metabolism, and overall vitality. The microbiome residing there determines clinical outcomes in ways we are only beginning to understand. Today, I will translate cutting-edge microbiome science into practical strategies you can implement immediately. Every day in practice, you see patients with chronic diarrhea, food sensitivities, inflammatory bowel disease, or unexplained immune dysfunction. What connects these seemingly different conditions? Dysbiosis, an imbalance of the microbial community in the gut. In humans, we carry at least 160 different bacterial species with over 536,000 bacterial genes, more than 20 times the host genome. In dogs and cats, we are discovering similar complexity. These microbes are not just passengers, they are active participants in health and disease. Understanding the microbiome transforms how we approach gastrointestinal disorders. Today, we'll explore the science, the clinical implications, and evidence-based interventions including probiotics, prebiotics, symbiotics, and postbiotics that restore balance and optimize gut health. The stomach's harsh and acidic environment with pH around 2 to 3 limits bacterial colonization. Only acid-tolerant species survive here. We see approximately 100 colony-forming units per milliliter dominated by lactobacilli and streptococci. As we move through the small intestine, bacterial density increases from duodenum to ileum, pH rises, transit time slows, and substrate availability changes, creating niches for diverse species. The small intestine is where most nutrient digestion and absorption occur, so the microbial community here must be carefully balanced. The colon is where microbial density reaches its peak, 1 trillion bacterial cells per milliliter. Over 90% are obligate anaerobes that thrive in the oxygen-free environment. This is the primary site of bacterial fermentation, producing metabolites that profoundly influence host health. The metabolic function of gut microbes is extraordinary. They ferment non-digestible dietary components like fibers, resistant starches, oligosaccharides that the host's enzyme cannot break down. Without microbiome, we would excrete these valuable nutrients. Bacterial fermentation salvages energy and produces essential vitamins. Butyrate alone provides up to 70% of the energy for the colonocytes, the cells lining the colon. This metabolic partnership is fundamental to the gut health. The presence of gut microbiota drives the gastrointestinal tract maturation. Germ-free animals raised in sterile environment without any bacteria show dramatically underdeveloped intestinal architecture. The villi are shorter, crypt shallower, mucous layer thinner, and immune system immature. Microbes signal the intestine to develop properly. They influence villus architecture, stimulate stem cell proliferation in the crypts, promote blood vessel formation, and most critically, drive maturation of gut-associated lymphoid tissue, GALT, which comprises 70% of the entire immune system. The protective function is multifaceted. Beneficial bacteria physically occupy attachment sites, preventing pathogen colonization, what we call as competitive exclusion. They produce bacteriocins that kill specific pathogens. They strengthen the mucous barrier. They stimulate antimicrobial peptide production. They train the immune system to distinguish friend from foe. Butyrate is remarkable, the preferred energy substrate of the colonocytes, accounting for up to 70% of their total energy consumption. But it is more than fuel. Butyrate has potent anti-inflammatory effects, strengthens tight junction, reducing permeability, and regulates cell proliferation, preventing abnormal growth. All three short-chain fatty acids also lower the luminal pH, creating an acidic environment in the colon that inhibits pathogenic bacteria while favoring the beneficial species. This pH shift is a simple but powerful protective mechanism. Not all bacterial fermentation is beneficial. When undigested protein reaches the colon, especially in excess, bacterial fermentation produces putrefactive compounds that can negatively impact the gut health. This is why dietary protein quality and quantity matter. Highly digestible proteins are absorbed in the small intestine, never reaching the colon. But poor quality protein or excessive protein intake beyond the digestive capacity pass undigested to the colon, where putrefactive fermentation produces certain harmful compounds. The goal is to maximize carbohydrate fermentation, producing beneficial short-chain fatty acids, while minimizing protein fermentation, producing putrefactive compounds. This is achieved through balanced, highly digestible diets with appropriate fiber content. In health, there is an elegant organization. Bacteria in their place, barrier intact, immune system vigilant but calm, beneficial metabolites being produced. In dysbiosis, this organization collapses. The barrier breaks down, bacteria and their products translocate, immune system overreacts, oxidative stress damages tissues, and a vicious cycle perpetuates chronic disease. This is the pathophysiological foundation of inflammatory bowel disease, chronic enteropathies, and many other GI disorders. Understanding this transition from eubiosis to dysbiosis is essential for effective therapeutic intervention. Chronic enteropathy in dogs and cats represents a complex syndrome. While not completely understood, it develops from multiple interacting factors. Individual patients respond differently to these interventions. Some dogs improve with diet alone, others require immunosuppression. But increasingly, we recognize that antibiotics and immunosuppressives, while controlling the symptoms, may negatively affect the commensal microbiome, potentially perpetuating the underlying dysbiosis rather than resolving it. This is why microbiome targeted therapies, the biotics, are gaining importance. They address the root cause, the microbial community imbalance. These 6 hallmarks don't occur in isolation. They are interconnected. Pathogen overgrowth damages the barrier. Impaired barrier increases permeability. Increased permeability activates the immune system. Immune activation creates inflammation. Inflammation further damages the barrier. It's a vicious, self-perpetuating cycle that requires targeted intervention to break. Probiotics are defined as live microorganisms that, when administered in adequate amounts, confer a health benefit on the host. Prebiotics must meet 3 important criteria: resistant to gastric digestion, fermentable by intestinal microbes, and selectively stimulate beneficial bacteria. Symbiotics combine probiotics and prebiotics. The prebiotic fits the probiotic specifically, enhancing survival and colonization. 1 + 1 = 3. Postbiotics are preparations of inactivated microorganisms or their components that still confer health benefits. Even though not alive, bacterial components are recognized by the immune receptors, modulating responses and supporting barrier function. Restoring gut health requires more than just adding probiotics. It demands a comprehensive approach addressing multiple aspects of gastrointestinal function simultaneously. Beneficial bacteria, their food source, anti-inflammatory support, and complete and balanced nutrition. Godrej Ninja simplifies clinical implementation of the microbiome science. Rather than prescribing multiple separate supplements like probiotics, prebiotics, omega-3s, antioxidants, the practitioners have one complete solution that addresses gut health comprehensively. The high-quality protein minimizes the undesirable colonic protein formation. The 3 P's restore microbial balance. The omega-3s reduce inflammation. The micronutrients support barrier function and immunity. This integrated approach does not just manage symptoms, it addresses root causes. By restoring eubiosis, reducing inflammation, and supporting barrier integrity, we create conditions for sustained gastrointestinal health. For chronic enteropathy patients, food-sensitive animals, or those recovering from GI diseases, comprehensive nutritional support with Godrej Ninja provides the foundation for recovery and long-term wellness. The gut microbiome represents a paradigm shift in veterinary medicine. We are no longer just treating symptoms, we are restoring ecosystems. Dysbiosis underlines countless gastrointestinal disorders, from acute diarrhea to inflammatory bowel disease. But with evidence-based interventions, probiotics, prebiotics, polyphenols, and comprehensive nutrition, we can break the cycle of inflammation, restore microbial balance, and transform patient outcomes. Remember, the microbiome is not just bacteria, it's a functional organ influencing immunity, metabolism, behavior, and overall health. Invest in gut health, and everything else follows. With proper understanding and targeted intervention, every patient can achieve optimal gastrointestinal function and vitality.